Hello everyone, I'm Ajay Tadinada, a junior at Nueva High School. My project is about unraveling the unknown mechanisms of macrolides to help address mucus hypersecretion in inflammatory respiratory diseases like COPD, IPF, and asthma. I'm basically trying to figure out the exact human binding target that is enabling the therapeutic behavior of macrolides to suppress mucus hypersecretion. The project included three key steps and will be followed by a wet lab post-pandemic. The first aim of the study is to generate biologically relevant 3D structures of macrolides and identify flexibility sites through all atom molecular dynamics. As you can see on the macrolide panel derived from MD simulations, there are clear differences in flexibility and structure across active, inactive, and control macrolides. You can also see an example of a 1 nanosecond of azithromycin solvated in water. The second aim of this study is to inverse dock these energy minimized 3D macrolides against the 17,000 SCPDB druggable binding sites. The results obtained from the inverse screen were analyzed on the basis of binding energy scores and the pocket fit of the macrolide, which can be seen on the screen above. The third aim of this study is to take these top 10 proteins and narrow them down to the top 3 candidates using molecular dynamic simulations. Here you can see the procedure that has been followed. The system is being loaded and built by adding a membrane to the protein and is then further solvated by TIP3P water models. Here you can see an example of a protein undergoing molecular dynamic simulation, in this case PDB-ID 4U8M, complex with macrolide changing its conformations during each frame. Analysis of each frame was done on the basis of RMSD and RMSF of the protein ligand complex. The results concluded that oxidoreductase enzymes have the most potential target binding sites. In terms of future steps, these three shortlisted oxidoreductase protein macrolide complexes will be subjected to perform wet lab studies to determine their efficacy and activity for mucus hypersecretion. Overall, this has been an exciting journey with a lot of learning. I look forward to the feedback and continuing to work on this project. Thank you for listening.